Hello, everyone. My name is Rodney Wallace. I am the Director of the Detection, Diagnostics, and Devices Infrastructure Division at BARDA. And welcome to our breakout session for today. I'll be giving everyone an overview of the objectives and accomplishments of the division, followed by Dr. Daitch, who will provide some key initiatives going forward. Then we'll have a product showcase. So a number of our project officers and contractors will get up and present some details about some of our programs that we think are representative of the breadth of our portfolio, and of course are things we haven't highlighted before. Then Mr. Urias will present a primer on how to prepare a successful white paper, and then we'll have a question and answer session. So one thing I want to just get straight in everyone's minds from the beginning is that back in 2020, BARDA and DDDI's world changed in a big way. We had to focus everything, all the resources we had on COVID response. And so we had to put all matters that were not COVID related on hold. In some cases that included submissions that had been in process for 6 months or more. We focused on COVID matters. Projects or contracts that had already been awarded did continue to proceed as best they could with much less BARDA support. And we proceeded forward to develop diagnostics and other important things for COVID. Toward the end of FY21, we were able to return to those submissions that were put on hold and start resolving those, and I want to thank the contractors who were affected by that pause for their patience. I know it was very frustrating. I'm happy to report, however, today that we have worked through all of those submissions and all those matters are resolved either with a contract award or with a letter that says thank you for submitting, but no thanks. Early in FY22, we were able to reopen our CBRN and influenza areas of interest in the broad agency announcement and start accepting submissions. And then later in FY22, we closed our COVID-19 areas of interest simply due to a lack of funding. Today and going forward, we're returning to our roots. We're certainly continuing to execute our COVID-19 matters. We still have a couple left to award. But our main focus for a while now is going to be our CBRN and flu areas of interest, back to BARDA's roots. So DDDI develops diagnostics across all of BARDA's portfolio of threats. For CBRN threats, we develop biodosimetry tools, biothreat diagnostics, and diagnostics to inform the appropriate use of antimicrobials. For influenza and emerging disease threats, we develop influenza diagnostics, emerging disease diagnostics like Zika, like Ebola, and of course COVID-19, and respiratory protection devices. BARDA's COVID-19 response for diagnostics, we had more than 500 industry engagements. We've achieved 1 EUA so far on a BARDA-funded diagnostic. We have 9 510Ks in review at FDA for BARDA-funded COVID diagnostics, and we received 29 EUAs. We also funded manufacturing capacity expansion or onshoring at 6 facilities in the country, 4 of which are complete at this time. If you look across our entire portfolio, and this slide's talking mostly about FY22's activities. At the end of the year, we had 68 products in support, 40 of which were lab, 20 of which were point of care, 20 of which are home use. We had 1 510K clearance, 7 510Ks in review across the portfolio. I'm sorry, that's incorrect. 13 510Ks in review across the portfolio. We received 3 new EUAs in 2022, and we still have 6 in front of FDA for review. Some of the lessons we learned in COVID. First, we need to improve our solicitations and our contracting mechanisms and processes. We have to be able to move faster. Dr. Dispro discussed that in his opening remarks this morning. It's a BARDA-wide mission to make that better. We also need to increase the size of the DDDI team. We approximately doubled during COVID, and we're not going back down. We're staying as big as we are. We also need to establish standing agreements with key diagnostic players that have facilities and capabilities to come to bear in a pandemic. It's just no reason to be negotiating new contracts in that scenario. We need to have agreements in place. And that'll be part of our improved contracting approach. We also need to continue increasing domestic manufacturing capacity and maintain the capacity we have. Our ability to do that is certainly going to be throttled by availability of funds. As Gary mentioned this morning, the White House issued an update to the National Biodefense Strategy. It establishes the path that the U.S. government envisioned needs to be followed to prepare for the next major pandemic. I encourage everyone to download a copy of it. Don't just read the first 3 pages. Go back into the attachments where all the good information is, and you can get a real feel for where the government is headed. 3 of the areas that are very important to DDDI are summarized in the slide. And the important thing to notice is the short time frames for those products to become available. So there's going to be a lot of interest in products that can act quickly, become available quickly, and be deployed quickly in any future pandemic. And Dr. Daitch will go into that a little further in a minute. So the outlook for DDDI. First of all, near term, we're going to finish our COVID response work. Many of those efforts are going to go all the way through 510K. It'll take us a number of years. As mentioned before, we're returning to our roots, CBRN and influenza threats, and we're going to complete our ongoing manufacturing capacity expansion programs. In the midterm, we're going to continue to focus on making molecular performance diagnostic tests available at the home and in remote settings. And, as I mentioned before, we're going to focus on a rapid contracting mechanism. Longer term, as defined in the National Biodefense Strategy, we're going to develop threat agnostic tests and viral family diagnostics that can be used in those early days of a new pandemic outbreak before more traditional threat-specific tests can become available. We're going to focus on filling that gap at the beginning. We'll also look at technologies to speed up the development of the more traditional diagnostic approaches. We also want to expand and maintain domestic diagnostics manufacturing capacity. So next up is Dr. Charles Daitch, who will give you some information on key initiatives. Chuck?
Great. Thank you, Rodney. Good afternoon. It is my pleasure to walk you through DDDI's key initiatives in greater detail. Next slide. Aha. Very good. As Rodney highlighted in his presentation, our partnerships made a significant impact on the COVID-19 pandemic, as highlighted by these 29 EUAs. Our team is working to support these projects through final 510K clearance to ensure these diagnostics remain accessible to the American public. Recently, DiaSorin Molecular was granted a FDA clearance for its SARS-CoV-2 molecular test. Next in our queue, there are 9 more BARDA-funded diagnostics in review with the FDA. These regulatory successes represent diagnostics across the entire continuum of care, including the traditional laboratory and point-of-care testing platforms, and includes affordable testing, which is closer to the patient, such as physicians' offices, tribal facilities, field labs, and homes. We are applying the lessons learned from COVID and expanding our platform capabilities to target IEID, influenza and emerging infectious diseases. Our efforts will include multiplexing influenza and influenza RSV assays to a number of COVID platforms. Adding influenza will significantly expand the utility of these platforms and ensures that they will remain competitive in the market. The systems include lab, point-of-care, home use diagnostics, and include a new non-invasive sample collection device. A few key requirements you should take note of are products which are low cost, rapid, home use, can differentiate influenza A and B, and include integrated patient management solutions. In addition, we are also looking for diagnostic assays for seasonal influenza, human-animal reassortment influenza, and emerging respiratory viruses. Our other major key initiative area is for CBRN threats, to include biothreat agents and AMR, antimicrobial resistant bacteria. Our focus with the AMR portfolio is to inform effective prescription practices for valuable antibiotics and provide an effective means to practice stewardship without endangering patients. Our broad portfolio includes molecular and sequencing-based AST, antibiotic susceptibility testing, host response viral versus bacterial assessment, and combined phenotypic identification with AST. We are well on our track to meet and exceed these goals outlined in the BARDA strategic plan, with one product 510(k) currently in review and 6 more working towards a submission. While there is a great body of work being undertaken, we are still looking to fill gaps and increase our shots on goal for diagnostics to inform antibiotic usage. The other area of interest under CBRN is the development of diagnostics for the detection of biothreat agents, such as those shown on the bottom right of this slide. Our biothreat diagnostic platform portfolio includes high-throughput laboratory and point-of-care diagnostics, with 3 platforms in 510(k) review and 5 more on the way. In general, we are looking to leverage existing adaptable platforms to address biothreats of interest. While our main focus is to develop on assays on smaller systems, we are also interested in the high-throughput assays. A key point for each is that we want to support platforms that aren't a one-trick pony and have the staying power in the market independent of government support. I wanted to highlight for you a new area of interest for a field-usable molecular filovirus test. Health providers in the field need to be able to detect and ideally differentiate between Ebola species, such as Zaire and Sudan, as well as Marburg virus, because the vaccines and therapeutics have limited, if any, effectiveness across these viral species. And while we have portable antigen tests for post-mortem detection, they are not specific enough to inform patient care and help curb any outbreaks. Molecular assays are well suited to this aim, and we have the opportunity to leverage the recent advances in small, portable molecular platforms that were developed as part of the COVID response. Next, I wanted to highlight an internal initiative we are working on, which may be welcome news to you regarding our ability to execute contracts more efficiently. During an emergency, time is of the essence to identify products that meet our requirements and secure funding contracts as quickly as possible. Our traditional mechanisms of BAAs, OTAs, IAAs, or service contracts are not as efficient as we want them to be. Our teams are diligently working to streamline existing solicitation processes and to develop new mechanisms. A good example of BARDA streamlining a current process is with our Easy BAA. An example of a new future mechanism would be BARDA establishing standing agreements with key partners. If you want to dig into this a little further, search beta.sam.gov for a sources sought notice entitled BARDA Rapid Response Partnering Vehicle. As outlined in the BARDA strategic plan, we are focused on accelerating development of emerging technologies that move testing closer to the patient and facilitate testing in limited healthcare resource settings. BARDA is supporting innovative research and development of home tests and wearable devices that will empower an individual outside of traditional healthcare settings. In addition, our goal is to rapidly deploy these after a new pathogen is identified. The faster we can diagnose, the faster we can treat and ultimately contain the spread of the disease. Take note that these will require minimal training and must be easy to use, have easy sample collection, minimal sample processing, and onboard reagents. In other words, you've heard us say this before, we need you to bring the complete sample-to-answer solution to the table. Overall, we are looking for accurate and affordable platforms to increase access for the population, and ones which are easily and rapidly adaptable to new threat targets in the event of future health emergencies. Shown here are some examples of successful adaptations of platforms when the new SARS-CoV-2 threat came online. BARDA was funding Q and Lucira for influenza prior to the pandemic, and we were able to leverage options to pivot these assays toward the COVID response. Q was the first at-home molecular platform to receive EUA and has a de novo application in FDA review and is currently working with BARDA on developing multiple new products. And Lucira has since achieved their EUA for SARS-CoV-2. As Rodney mentioned earlier regarding the recently published National Biodefense Strategy, in the long term, we are moving towards broad coverage tests, which can be used in the earliest days and weeks of an outbreak. The best way to think about this slide is to key in on the words broad coverage, instead of analyzing a patient sample and saying it contains the specific pathogen, SARS-CoV-2, Omicron variant, BQ.1.1. Alternatively, we are trying to get an idea if a patient sample contains a certain viral family, like the coronavirus family or filovirus family. The goal of threat agnostic is to detect every known pathogen from a single sample, whether it's viral or bacterial or fungal. And as a stretch goal, we would also like to be able to detect unknown pathogens. The idea here is, instead of looking at a biothreat from a list-based perspective and asking, what specifically is it? Is it and instead, we're more broadly asking the question, is it harmful, and how harmful is it? These types of diagnostics are poised to provide the tip-of-the-spear response in identifying new and emerging infectious diseases before a pandemic takes hold. They will fill critical patient testing gap in the first days and weeks, while the pathogen-specific diagnostics are developed in the first months. However, there are significant gaps that still need to be bridged, including technology and regulatory challenges. Our division has partnered with DRIVE to explore early development efforts of these products to help bridge these gaps, and particularly establish a regulatory framework. Moving on to manufacturing scale-up. The pandemic highlighted a critical need to rapidly expand production of diagnostic tests to meet the high testing demand. At the beginning of the pandemic, BARDA funded 6 companies, which range from small to large companies and include lab-based to point-of-care platforms, and molecular to antigen or antibody tests. Together, the companies have the ability to increase diagnostic test production by over 4-fold, totaling 72 million tests per month for current and future pandemic support. We have successfully completed 4 of the manufacturing expansion efforts. In the future, our goal is to have partners maximize supply chain efficiencies and to be able to produce on demand in order to eliminate or minimize the need for stockpiling. We also refer to this as maintaining a warm base. Companies who have solutions to these requirements will be in a great position to be competitive for future funding. Please note, we currently do not have any solicitations open at this time for capacity expansion awards, but keep your eyes open on the beta.sam.gov website for any future solicitations. So I wanted to finish up with a strategic topic. It may go over my time a little bit, but I think it'll be fun. So let's do it. You have likely seen in mine and Rodney's presentation some opportunities for funding, which could help your company's business. You also may have skipped over areas which we need help with, but don't look as attractive from your business standpoint. I encourage you to take a step back and look for creative solutions of achieving a win-win for your company and the government. For example, one advantage of partnering with the government is you can get non-dilutive dollars for both product and process development. One solution to think about is for you to try and design and develop your product and production processes so that you can leverage your commercial and government business lines off of each other. For example, let's say the government has a need for biothreat assays that are run on a field-portable handheld instrument and are low cost. Take a step back, take a close look at those requirements and explore if your company could be more competitive in your commercial markets with a version 2 miniaturized instrument that could move your commercial assays from, say, the lab into a doctor's office, or even better yet, for home use. Or could your manufacturing processes be improved to be more efficient so as to lower your cost of goods and increase production volumes? If yes, then you may want to consider taking a second look at biothreat assay development. Your creativity will come into play by making sure that both the biothreat assays and your commercial assays are able to run on the new version 2 instrument, and that both can be produced with the new production processes. Now you have brought a win-win solution to the table for your company and the government, and you have offset your IR&D budget with non-dilutive government funding. And if that doesn't entice you enough, the icing on the cake is the support you will get by BARDA staff towards your regulatory clearance goals. As an in vitro diagnostic developer, one of the hardest things you will ever do is navigate the FDA regulatory clearance roadmap. Trust me, I've successfully done it, and it nearly killed our company. If you become a BARDA contractor, we don't just write you a check and disappear and let you stumble through what we call the commercial valley of death. Instead, you will have access to BARDA technical staff, who are some of the best IVD scientists and engineers, the best clinical, regulatory, quality, and manufacturing experts I've ever seen in my career. Together, our BARDA staff will work hand in hand with your company to help get you over the commercialization goal line. We will not be happy, and we will not rest until your product is out there saving lives. In summary, another way for you to think about this is that BARDA prefers long-term strategic partners, and one way to form long-term relationship is to get your foot in the door and prove that you and your product can get it done. I am suggesting to you that you may want to revisit some of the AOIs you overlooked, because that may be the best way to get your foot in the door and provide a win-win. It doesn't matter if you're a large public company, a small private company, or an early stage company working out of your incubator or garage. I encourage you to throw your hat in the ring. If you look across our portfolio, you'll see that BARDA has some awesome success stories and long-term partnerships across all of these different size companies. So I thank you for your attention. If you like these types of strategic topics, make sure you stick around and attend the industry panel discussion being hosted by Rodney Wallace tomorrow. Up next, we have our project showcases highlighting product examples across our key initiatives and include products which have not been highlighted at BID in several years. First up is Dr. Davenport, a project officer in DDDI, here to tell you about our collaboration with Becton Dickinson. Matt, over to you.
Thank you, Chuck, and good afternoon, everyone. The goal of our partnership with Becton Dickinson Company, or BD, is to develop and obtain FDA approval for a number of tests that will detect and distinguish multiple respiratory pathogens from a single sample, as well as operate on 3 key BD platforms across the spectrum of healthcare. These include laboratory settings, point of care, and at home. Of these tests, one of the most interesting is a pan-coronavirus test that has 4 key attributes. The first is that the test will detect and distinguish known respiratory pathogens. These include the known coronaviruses, flu A, flu B, and RSV. Second, the test will be able to detect and distinguish or detect pan-coronavirus or novel coronaviruses that could lead to the next outbreak or pandemic. Third, the test will operate and be cleared on 2 BD platforms for use in the laboratory. This is the BD Max and the BD Core. And then last, and probably most importantly, is once the test is cleared, it will provide a capability that can be deployed rapidly in the initial stages of an outbreak before agent-specific tests are available. In addition to the pan-coronavirus test, BD is developing 2 other types of respiratory viral pathogen tests. The first will be a molecular test cleared on both the BD Max and the BD BD Max and the BD Core for laboratory use, and this will detect and distinguish SARS-CoV-2, flu A, flu B, and RSV. The second, in contrast, is an antigen detection test based on BD's Veritor platform. That will be able to detect and distinguish SARS-CoV-2, flu A, and flu B. In all, the BD partnership has at least 3 goals. The first is to provide critical testing for respiratory illnesses across the multiple levels of healthcare spectrum to help manage patient care. The second is to address novel coronaviruses of the future that may lead to the next outbreak. And the last is to meet a critical goal to make testing more accessible and available, particularly at point of care and at home. Next up is our fellow project officer, Eun Mi Lee.
Thank you, Matt. Hello, I am Eun Mi Lee, and I'm going to talk about the partnership with DTECT, which started earlier this year. DTECT will advance the development of a next-generation device that detects COVID-19 and flu tests through FDA EUA and 510(k) clearance. It is a multiplexed molecular platform that identifies and distinguishes influenza A and B, as well as SARS-CoV-2, from a single nasal swab. Individuals can perform this test at home without requiring laboratory expertise or sophisticated equipment. It includes a disposable swab, a single-use cartridge that you can see to the right of the swab, a reusable hub, and a mobile app. The mobile app will provide instructions for the user to run the test. The COVID-19 and flu test uses isothermal amplification and real-time fluorescence. The sensitivity and specificity is comparable to PCR, with results in less than 30 minutes. Results are received from the hub to the mobile app via Bluetooth. The app will also help with interpretation of results. The COVID-19 and flu test uses enzymes developed by DTECT and will be low cost. The platform can also be expanded for additional infectious diseases. Thank you for your time. And next is Dr. William Kramp, a senior diagnostic advisor to DDDI, who will talk about CLUX.
Good afternoon. CLUX has been partnered with BARDA since 2018. This program falls into the category of Area of Interest 7.3, antibiotic resistance diagnostics for priority bacterial pathogens. Pre-COVID AOI 6.8.5 expanded on this, looking for phenotypic platforms and assays for moderate and high complexity laboratories, assay time to identifying resistance or susceptibility to irrelevant antibiotics in less than 8 hours, and high throughput. The CLUX program consists of 3 phases: Gen 1, Gen 1.5, and Gen 2. Gen 1 consists of an inoculation station and an incubator. Samples from colonies are dispensed in 384 well plates, which contain a full dilution series through breakpoints for all of the major classes of currently used antibiotics. There is available space on these plates for antibiotics as they are developed. Resistance is determined by cell shape change of the bacteria in the presence of the antibiotics. The incubator can hold 96 384 well plates, and the combined time to result, including inoculation, is approximately 5 hours. This makes the time from patient to AST result 36 hours, which is an improvement of the current standard of care, which ranges from 48 to 96. Both gram-positive and gram-negative versions of this test are currently under 510K review at FDA. Gen 1.5 is a positive blood culture processing unit, which once a culture is flagged as positive, this unit processes the sample to a multi version, which can then be used for identification and then also be passed to the inoculation station. This process takes us less than 1 hour. A pre-sub has been submitted to FDA for this new sample type. If successful, this would reduce patient to AST results to 24 hours, which is concordant with the 4th empiric dose of a normal treatment course. Gen 2 capitalizes on the current spoke and hub model of AST testing. Spoke labs would send their samples to the hub in mini incubators, which monitor growth of the blood culture. The hub would then put these mini incubators in a docking station, continuing monitoring. When they became positive, they would then be sent to the blood processing unit and off for ID. Benchtop feasibility studies have shown that this all major positive bloodstream bacteria can have a full AST profile done within less than 16 hours. This would represent a significant improvement in patient care and antibiotic stewardship. I will then turn this over to Dr. Molly McLendon, who will talk to you about BioFire.
Good afternoon. I'm Molly McLendon, and it is my pleasure to tell you about a newly formed relationship between BARDA and BioFire Defense. BioFire Defense Warrior Panel is a collection of molecular nucleic acid amplification assays that detect the biothreat pathogens that cause anthrax, tularemia, plague, Q fever, Ebola virus disease, and Marburg virus disease. Depending on the pathogen, the panel can detect organisms from whole blood specimens, blood cultures, and sputum specimens, and the test provides results in 45 minutes to an hour. A de novo request was granted by the FDA for the Warrior panel using whole blood specimens and blood cultures in early 2017, and the test received 510K clearance a few months later to use sputum samples. The panel is currently FDA cleared for use only by the Department of Defense and is run on the BioFire's FilmArray 2.0 instrument. On the right side of this slide is the BioFire FilmArray torch system. This instrument is capable of higher throughput than the FilmArray 2.0 instrument because it can run up to 12 samples at a time. It is also randomly accessible. Under this new contract, BioFire Defense will advance the development of the Warrior panel and a panel of control assays for use on the higher throughput FilmArray torch instrument. Once these bridging studies are complete, BioFire Defense plans to apply for a 510K clearance from the FDA for both of the panels on the torch instrument. The intended use of the Warrior panel is for individuals with signs and symptoms of infection from biothreat agents, or individuals who are at risk for exposure or suspected of exposure to these agents in response to a confirmed or suspected bioterrorism event or outbreak. Clearing the Warrior panel for use on the FilmArray torch system will significantly increase the U.S. national biothreat testing capability in response to an emergency declaration and helps bolster national preparedness capabilities to protect people from deliberate or naturally occurring outbreaks of biological threats. I'd like to thank you for your attention, and now I will pass the presentation over to Chris Knickerbocker.
Thank you, Molly. Good afternoon, everyone. Today I will be discussing Senso Incorporated, which is a global life science company developing high-accuracy, low-cost point-of-care diagnostic technologies with locations in Newtown, Pennsylvania, and the United Kingdom. Their current partnership with BARDA began recently in September 2022. Senso has developed a high-sensitivity COVID-19 amplified lateral flow rapid antigen test. This test is a rapid, single-use lateral flow immunoassay for the qualitative detection of SARS-CoV-2 S1. The Senso test uses a novel, proprietary method to bind detection reagent and capture antibody in the test zone of the cartridge. Since the Senso detection reagent will only remain bound to captured antibodies in the test zone of the analyte is present, it can be amplified without fear of amplifying noise and therefore generating false positives. If the target analyte is not present, the detection reagent and capture antibody will flow completely off the strip into an absorption pad, and amplification reagent can be safely applied without impacting specificity. The Senso test has PCR level sensitivity and accuracy and can detect approximately 10,000 times lower than current lateral flow tests. The test gives results within 7 minutes, utilizing a visual readout with no specialized equipment and/or training necessary. Nasal swabs are used for collection, so ease of use is there for home testing. And the test targets an LOD of approximately 20 copies per mil. Senso will be seeking an EUA for home use at the end of this contract. And lastly, the Senso ALF platform has potential use across a wide range of infectious diseases and targets other than COVID-19. These include, but are not limited to, TB, Hep C, HIV, flu A and B, and a number of STD organisms. Thank you for your time and attention. And next, I would like to introduce Dr. Dave Mittar, a project officer in BARDA Drive.
Good afternoon. My name is Dave Mittar. I'm part of Drive Division at BARDA. The project that I'm going to present is part of our next generation sequencing-based agnostic diagnostic portfolio. In the next 2 minutes, probably I'll be using this agnostic term for at least 10 times so that you make sure you understand that the goal of this program is to develop a threat agnostic platform that should be able to detect any and every respiratory RNA virus for future pandemic preparation. Plus, the goal is to commercialize this technology so that it can be used in case of any future pandemic, as it can detect any novel and emerging viruses as well. We have funded 5 partners in this agnostic diagnostic program, and all of those 5 partners are addressing the challenges that are hindering the commercialization of this technology. Some of the challenges include host background, DNA, RNA, optimization of assays, analytical clinical validation, clinical grade bioinformatics, and on top of that, the regulatory clearance for these assays. And all of our 5 partners are addressing these challenges. Out of 5 partners, 3 of them are from academia, UCSF, UCLA, and Columbia, and 2 of them are from industry, Jump Code Genomics and BugSeq. The project that I'm going to present is from Columbia University. What they have is a agnostic assay called VirCap-seq. And the uniqueness of this assay is that it uses a capture probe technology where you can enhance the sensitivity assay equivalent to existing RT-PCR or other molecular assays. These capture probes are known to bind any and every known vertebrate viruses with up to 50% homology, which means that this assay should be able to detect any new and emerging viruses with at least 50% homology. And they recently got the CLAP certification for their assay from New York State Department of Health. And in the project with BARDA, they are doing analytical and clinical validation of their assay on Illumina platform. Their early data is very promising, where they've shown LOD up to 5 copies per mil in a dozen of viruses. And they have also submitted a pre-sub to FDA for seeking regulatory guidance. Eventually, they will be doing clinical validation as well And lastly, they would also be performing proof of concept study on long-read sequencing platform that is Oxford Nanopore technology. Thank you very much for your attention. And next up is Dr. Adrian Urias, who is our SME for medical devices. He's going to talk to you about some tips and tricks for successful submission of your proposals to us. Adrian.
All right, thank you, Dave. So the first point that I want to make is for any successful submission to BARDA, understand that DDDI funds product solutions that are ultimately regulatory cleared. We are not looking to basically incrementally improve upon a technology. So write your submission accordingly. So some critical elements that you want to include in your submission are listed below. First is the area of interest. Tell us which one you're applying to so we know what to review you against, because each one has its own set of definitions. Don't make us guess what we have to grade you against. Give a technical explanation of your product and your project development. This is not a high-level sales pitch. This is more of a peer-reviewed publication. We're going to have multiple subject matter experts looking at your paper. As such, be sure to include a lot of supporting data. So we don't want to know just about your idea, but how does it work and how well does it work up to this point? Clinical utility. Is there a market for this? Ultimately, as Chuck mentioned earlier, the government does not want to support by buying your product at the end of the day. We want to see that there's a market for it, so it's self-sustainable. As part of your submission, be sure to include pictures of your prototype. We know those don't always look the best, but that also tells us where you are in your project development. And we're familiar with seeing that, and that gives us an idea of how the device works as well. And be sure to include any challenges and risks for the project development, because we understand that happens. If you don't include those, that makes us wonder if you actually understand the development process and what you need to achieve ultimate success. So I want to go over some common pitfalls that we see in a lot of submissions. So first off, if you've ever had a tech watch, a market research call, or even if you already had a white paper submission, do not assume that your reviewers are going to be familiar with your company or your product. So your submission should be a standalone product that stands on its own for review processes. Especially for white papers, do not waste space telling us how bad the problem is. We know how bad the problem is. We want to know what your solution is to that problem and what makes your device unique compared to what's already out there in the market. Don't expect reviewers to read any outside references. While you can put citations on there, don't include something critical that you want the reviewers to understand in those outside references, as they are not required to review those. There's also, in the broad agency announcement, there's an appendix that actually shows you the judging criteria that your product and project will be judged against. So you actually have all three elements that you're going to be looked at, including all the subtasks within it. So be sure to look at that, because that's what we're going to be using to ultimately judge and score your submission. And another one that we often see is insufficient technology readiness level, or basically not mature enough. So we're going to go into a little bit more detail now. So TRL 1 or 2, that's usually your first idea, very early research, way too early for BARDA funding in our division. TRL 2 or 3, you might have some breadboard functionality. You're just starting to kind of prove out the idea. Still probably too early. TRL 4 or 5 is more of our sweet spot. That's where you already have some key product requirements. You developed your prototype, and you achieve some of the data to show that you can reach those goals. So there is feasibility and being able to show us that you can ultimately achieve what you're proposing to us. But keep in mind that each area of interest has its own TRL requirement. So again, be sure to look at that before you submit. As you get more towards the proposal stage, we're going to be looking at a lot more detail. So in not only the TRL worth in terms of your product, but also in terms of your company. So as you get further and further along in the TRL, we also expect you as a company to be able to accompany that with all the required documentation that goes along with that product development. So all of your requirements, all of your project reviews, all of your system integration, your V&V testing, we should be able to see that you understand all those different elements of development. Along with that, again, for the proposal, while you want to touch on this in the white paper, you don't have a lot of space. But in the proposal, you want to make sure that you cover the entire breadth of your capabilities as a team for that project. What other resources are you assigning to it? Who is your entire team for each different aspect? So not only your key personnel from the technology side, but what's your marketing plan? And do you have a clinical plan, a manufacturing plan, project management. All these are very important for us to be able to assess that, yes, not only do you have a good idea, but you have the team to ultimately be able to succeed in developing it to fruition. So just kind of taking a step back and looking at some of the big pictures, we recommend having someone else that didn't write the paper to actually review it, just for clarity. Does it read well? Do your figures, is everything labeled properly? Just to make it easier for your reviewers so we're not trying to guess and interpret what you meant to say. And be sure not to include any gaps in the project development, because if you go from one stage to the next without describing how you got there, that tells us that you don't really know what you're doing, and that's a red flag for us. So we want to see all the steps that are necessary to get to the end of the day so that we know that you can have a successful project. And going back again to utility, what's the market for this? Who's going to be using it? Why are they going to be using it? Why are they going to buy your product in place of another one? And what does your device cost? Are all these feasible? Is this in line? Are you ultimately going to be able to get this commercialized? And then realism. Don't be too optimistic and tell us you're going to do a 3-year project in 1 year because you think that's what we want to hear. At the end of the day, we're going to see that as a red flag as well, that perhaps you don't know what it's really going to take to develop this. So there, we expect to see multiple iterations of development. That's a normal process. So be realistic in whatever you propose. So to look at all of the solicitations, you can go to the website listed below at medicalcountermeasures.gov. At this point, I'm going to turn it over to Rodney for a final wrap-up.
Thanks, Adrian. All right, so first of all, thank all of you for your attention. At this point, you should have a feel for some of the directions of DDDI going forward, some information on some existing funding opportunities. Adrian provided some really good hints on how to prepare a successful submission to BARDA. And in a moment, you're going to learn how to reach BARDA if you have more questions. However, before I jump there, tomorrow we're having an industry panel with 7 leading industry participants to discuss topics about the diagnostic industry. We strongly encourage folks to attend. I believe it's at 1:30, though apparently I messed up the time of this session this morning, so I'm not going to guarantee it is at 1:30. But please join us. Also, we have virtual expo booths, 4 to 6 this evening, 9 to 10 in the morning, and 4 to 6 tomorrow, where you can actually talk with some of the DDI team members if you have questions or want to get more information. And here is how to reach BARDA. It's probably a little disappointing these are not people's email addresses, but that is because that allows people to actually, more than one person to answer your questions, so we are guaranteed to pick it up. So with that, I think Dr. Koh is going to join us here to lead or moderate our question and answer session. If you do have questions, please put them in the chat.
Thanks, Rodney. So apologies, everybody, for the technical difficulties that we've been experiencing, but unfortunately, we cannot see the Q&A live here in the auditorium, and I also can't see the chat box. So for any questions that you have, please go to our expo page. We'll be there answering questions this afternoon from 4 to 6, and also tomorrow from 9 to 10, and again from 4 to 6. Very sorry for that. We only have a few questions, or a few minutes, so I'll turn to a few of our frequently asked questions just to give folks a flavor of the questions we usually get, and some answers, hopefully that'll save you some time so you don't have to ask us later on. So Rodney, I'll turn the first question to you. What is the preferred method to ask questions to DDDI and to BARDA?
So hopefully, oh, it's not working.
Hello?
Oh, here we go. All right, so the slide that's up right now actually has 2 of the best ways to reach out to DDDI and our contracting shop for questions. You can also find that technical contact email in the solicitation. And again, the reason we want you to reach out to these non-human specific email addresses is so that more than one person can address your question.
Thanks, Rodney. Chris, what is the main difference between a market research call and a tech watch appointment?
Okay. There we go. Okay, a tech watch is a semi-formal meeting requiring a formal application. And if accepted, it will result in an, interagency government audience for you to present your technical data and for opportunity for Q&A. A market research call is usually more informal and meeting with specific BARDA teams such as DDDI or DRIVE. And again, you will present technical data and also have the opportunity for Q&A. A market research call usually stems from questions that one may have when reviewing the BAAs or easy BAAs and reaching out to the point of contact for those particular solicitations.
Great, thank you. Matt, so could you talk a little bit about what we've seen for some top reasons for white paper rejections?
Sure. Adrian gave an excellent overview of things to be careful of and to watch out for when you submit a white paper or proposal. Probably the number one thing that reviewers catch is whether or not your technology meets the technology readiness level, or TRL. So as Adrian covered, we typically fund TRL 4 and above. So when you submit your proposal, you should provide evidence that your technology actually is TRL level 4 or above. That would include, as Adrian pointed out, pictures, as well as data with your device or your test, and as close to the real sample as you can provide.
Okay, thanks, Matt. So Chuck, you talked a little bit about some of BARDA's interests in point-of-care technologies. Do we require that POC devices have a battery?
The first thing you're going to need to do is make sure the area of interest you're going into doesn't specifically say battery. If it's not there, then you have to go into the next level of kind of what I say putting your strategic hat on. So if you're going point of care and you're trying to set your plate to get out there and address the biggest customer base, what you can probably guess is by putting a battery, including that into your device, is going to increase your access to the market. It may be interesting from getting into a physician's office, but you don't know if something's going to come down the line and BARDA says right on the heels of that, we need you to get into a field lab, or we need you to get into the bush where there is no power availability. And right off the bat, you're up early. The early bird gets the worm. You're going to be first in line to say, I have the solution for you. So I would say, think about it.
Thanks, Chuck. So Molly, we often get some questions about whether or not there's a limit on the amount of money or the amount of time that our offers can request. Could you talk a little bit about that?
Yeah, so there is no limit for the amount of money you ask for, but we do understand how much the development of these tests cost. So asking for too little indicates either you're trying to impress us and lowball, and that's not going to be enough money to make a product that can be successful on the market. Alternatively, asking for too much is also problematic. Any contract we fund through the BAA has a 5-year period of performance limit. So the money has to work within that 5-year time period. During the COVID-19 response, we definitely gave out some very large awards. However, that's not going to continue. Instead, our awards are going to go back to smaller amounts of money that you can think of prior to the COVID-19 pandemic. Yeah.
Great, thanks, Molly. So that's about all the time that we have. So again, thank you all for your time and attention, and apologies for technical error or difficulties. So again, if you have any questions, please do feel free to visit us at our expo, either 4 to 6 this afternoon or tomorrow 9 to 10, and again at 4 to 6. Thank you.